Welcome to the ASGCT 2026 annual meeting in Boston. This week we are talking with some of our ASGCT members to learn more about their backgrounds and talk about the ASGCT membership. I'm here today with Marco Carpenter. Marco, thank you for coming to the show.
Yeah, no problem. Glad to be here.
Excellent. All right, we're just going to take you through a key set of questions that we've been taking everybody through. Can you give us a quick intro to your role and the work that you're focused on right now?
Yeah, of course. I'm a postdoctoral fellow at the Children's Hospital of Philadelphia, and my work sits at the intersection of neuroscience, pharmacology, and bioengineering. And the big picture goal is to develop gene therapies for devastating genetic disorders of the brain specifically. And so right now I'm focused on engineering lipid nanoparticles, or LNPs, to deliver therapeutic mRNA to specific cells of the CNS. And so a lot of my work right now is centered on two diseases, spinal muscular atrophy and Angelman syndrome. And I'm also running a number of base editing screens to really identify novel ways to treat these diseases.
Fantastic. And this is a question that's always interesting to ask. What first drew you into cell and gene therapy?
Yeah, so for me, I think it was the diseases themselves. So when you look at conditions like Angelman syndrome or SMA, it was really, you realize that the root cause is a single gene, and the idea that you could go in and perform molecular surgery and fix it at the root cause is what really led me to pursue gene editing and gene therapy.
Excellent. And what part of the space would you say has you most excited at the moment?
Yeah, so I did a lot of work in fetal gene editing or prenatal gene editing, and it's really the idea that you can, again, intervene before disease fully takes place and before neurons are lost or before a developmental window closes, and that's something that I find really exciting. And so my lab will also include in utero mRNA delivery for SMA and Angelman syndrome. And the results suggest there's a real potential there for fetal gene editing in these diseases.
Love it. Excellent. Yeah, I got a chance to hear a part of a presentation from one of your colleagues at CHOP making artificial wombs.
Oh, so you were Alan Flake. Yes,
Yeah.
Very cool. Lab next door. I mean, there's the idea that we could also be potentially delivering these therapies through the artificial womb, which is another kind of layer on top of already doing fetal gene editing.
That's awesome. All right, what challenge or opportunity in the field do you think deserves more attention at the moment?
Yeah, so I think we've focused a lot about extrahepatic delivery or targeting other organs like the brain, but we really don't focus on the specific cell types. And so I think when we're talking about delivering to the CNS, we really need to focus more on what cell types we're targeting and what is the level of targeting that we're at in a specific cell type.
Makes a lot of sense. All right, shifting gears here just a little bit. How has ASGCT membership helped you in your career?
Yeah, so I think this one was kind of the first where I've kind of seen how ASGCT has helped to uplift kind of trainees. And so receiving this award, I'm now, getting a lot of hellos and congratulations from people that I've never known before. And that's kind of led to a lot of networking opportunities. So
Wonderful.
I've seen a lot of how that networking can kind of help me propel my career forward.
Excellent. And why has ASGCT continued to be valuable to you as a member?
So the gene therapy field moves incredibly quickly, and so being able to come to one place and just get a breadth of research or a breadth of what's going on across many different aspects of gene therapy is why I find it valuable.
Right on. Well, Marco, thank you for coming on the show. Very excited to see the work that you continue to do. Definitely an emerging field that's going to be exciting to watch.
Of course. Thank you.